Okay. So, good afternoon. So actually, we are evening here in Hong Kong. So, okay. So, anyway, I say good afternoon. And first, I'd like to thank the conference committee's kind invitation. And my topic is about antiviral drug discovery based on plant resources against emerging viruses. And talking about the viruses, so we know that viruses will have a lot of viruses. So the viruses come and gone, and they have been with us all the time in our human history. And even individual of us has encountered many viruses in our lifetime. So they are probably our biggest enemies. And here, and I just give you some examples of the some viruses. Okay, so just in the last 100 years, if you look at the history of the human, so in 1918, the Spanish flu influenza virus killed is estimated about 40 to 80 million people, and the HIV. So that's discovered in 1981. So has been with us for 40 years and has killed 36 million people. So, and this swine flu. So that's also another virus. And from 2019 to 2010, it's also caused by H1N1 influenza virus. So that affected about 60 million people and killed about 12,400 people in the US. So that's called swine flu. So, okay. And the next year, we see the Ebola virus. So that's a lot of outbreaks for the Ebola. So the most devastating outbreak is in 19. Actually, it's not in 19. It's 2014. So that's all the outbreak that killed about 11,000 people and affected about 12 and like 28,000 people. So about half of them were killed by the Ebola virus. Okay, so the mortality rates for Ebola is very high. It's roughly is about 40 to 90%. Okay, so this is terrible virus, and Zika virus. So that also caused birth defects, infected roughly 1.5 million people in 2015-16 outbreak in the world. Okay, so there's and the dengue virus also infected like 4.4 million people in Brazil in 2019. The dengue also with us for many years. So, and here I give you a list. Okay, so this list of this virus, emerging virus in the last 100 years. The major virus. Okay, so also some other viruses. So here the table I want to tell you that the origin of the viruses. So if you look at this table, you will see that most of the viruses actually originated from the animals. So and only one case, the Zika virus is originated from mosquitoes, so transmitted actually by biting from mosquito. So, and the Spanish flu, so that's 1981 Spanish flu. So that's probably the later research have found that it's probably also originated from this like from birds. So it's killed, like it's affected actually 1/3 of a population, world population. So this very terrible virus. And some other viruses like Ebola probably originated from bats, and the apes, and the HCV and the HIV were originated from chimpanzees, and bird flu like H5N1, the bird flu. So that one have although not so many cases, but if you look at the death rates, that's all. Among the 861 affected people, so 455 died. So and so that's also terrible because the bird flu in research like in 2000, I think that 2000 probably 11 or 13. So there is some 2012, I think. So there is a lab in the Wisconsin. They actually altered the gene of this bird flu. So and they created actually variant of this H5N1. So that make it this very very much like can be airborne transmitted. So for this virus, so sometimes you see that when we do the research on the antiviral, sometimes very dangerous. So we must be careful to deal with this virus. So otherwise, if they are leaking, so that would be cause terrible, probably to the human society. Okay, and the current SARS virus. So we all know that this already, you see, already infected more than 200 million people, and more than 4.4, probably 4.4 million people has died from this the new coronavirus. So and do we have the treatment? So for these viruses, for some viruses we do have the treatment. So like but a lot of viruses, we do not have a very effective treatment or vaccine. So here are at least some of the viruses, and like H1N1 virus that caused the 1918 Spanish flu. So this, at that time, there is no vaccine available, and for the currently, so the later on, so our research have developed some drugs like Tamiflu in 1999, approved by US FDA. Also 2018, another drug approved by FDA for treatment of this H1N1 flu. For Zika, so this Zika, actually there's no vaccine available, also no treatment, although there are a couple of vaccines are in development. For Ebola, there is actually there's one vaccine available, so that's proved by US FDA in November of 2019, and also there is drug development. One drug development that's made from 3 monoclonal clonal antibodies, so that's also available, but the effective still need to be seen. So for this vaccine or drug treatment. For HCV, actually, it's very lucky actually. So that's 2013. So a company called Gilead, so developed a drug so called sofosbuvir, so also Sovaldi. So that's first they approved by FDA 2013. So this drug actually is curable. So you see, if you look at our medicine, we do not have many drugs. We call that curable. So actually, but this Sovaldi actually we can call it a curable drug, because if a patient, HCV patient, take like about 4 weeks, for 12 weeks actually. The 12 weeks of the drug, so the HCV virus can be actually eliminated from the body. So that's we call it curable. And but for HCV, we still do not have vaccine available. Now let's look at the HCV. Okay, so HIV. So this HIV is started in probably 1981, probably also before that, but they first discovered in 1981. Since then, actually a lot of drugs have been developed. About 24 drugs developed, and 23 combinations have been proved by US FDA. And here I show you one of this called Retrovir. So it's the first anti-HIV drug proved by US FDA. So this although for HIV I have a lot of drug available, but these drugs we cannot call it curable. They are not curable. They cannot eliminate HIV from the body of the patient. So the patient, HIV patient, have to live with this virus for the lifetime by taking this medicines. And for other viruses like H5N1 bird flu, we have a couple of vaccines, although not very efficient, but we have some also drug treatment like Tamiflu I mentioned earlier. So this can be also can be some effective, but not very effective. Okay. And for SARS 2003, the first SARS, so that's we did not have vaccine at that time. Also, we did not have a drug treatment for this SARS virus at that time. But the virus actually is very strange. So it mysteriously disappeared after six months. After 6 months, appearance of this virus. So nobody knows the reasons why this is gone. So probably it's a tight control, maybe. So that's so. But at that time, no vaccine, no drug, but the virus actually gone. But the second coronavirus, we are not so lucky. So the current coronavirus started from since December 2019, almost 2 years. We now have a couple of vaccines available, but we also have some drugs available, proved drugs like the first FDA approved remdesivir in 2020. So, but we still cannot get rid of this virus. The virus keep changing. The variant keep coming. So it's you see that we still need further drugs. We still need further vaccines to curb these viruses. So for me, I'm talking about this because I focus on the plant natural products. So I was talking about this plant natural products for their importance. So our drug discovery is based on plant natural products, and we give you a chart. This chart let you see this importance of natural products. So this summarized by Newman and Craig, published in 2020. So they come up with that in this paper they say that about 50% of all FDA and equivalent agency approved drugs were derived from natural products, actually, either directly or mimic or synthesis. So that's if you look at the antiviral drug, so that's a 33% antiviral drug based on a natural product. But if we exclude the vaccines, the number is go up to 63. So 63. So there's a number. So larger number, larger number of drugs actually derived from natural products. And for the plant natural products are also very important. So and plant natural products are actually a lot of drugs actually derived from plants. So why the plants are important? So here the two reasons I give you. So that plant species are very diversified, and plant compounds are also very diversified. So for the plants, if you go around the world, you will see that many different plants. So they actually you can find the approximately 300,000 higher plants in all the world, probably even larger number than this. So very diversified plants. So and for the okay, let me see that map. So just to give you some idea about this, how the distribution of the plant. So you see circled the 5 spots, hot spots. So here is a you can find a very diversified plants, and you can find about more than 5,000 plant species. It's just around say 10,000 kilometers, so square kilometers. So very diversified plants. You find this area. So deep color, you can find more plants over there. So and for the plants, for the plant compound. So actually we're talking about the diversified the plants. So not in terms not only the numbers. So a lot of compounds have been discovered from plants, but also in terms of the structures. If you look at, I gave you some examples here, the structures. So the taxol and the ginkgo biloba. So this triterpenes. If you look at the structure, they are very, very, very complex and contain a lot of chiral centers. So the structure much more complicated than what they synthesize the structure. So also, if you look like the plant extract, we can find like a lot of, if you do the HPLC analysis, we can find a lot of peaks over there, which means a lot of compounds over there. So plant extract actually contain probably more than 1,000 compounds, and many of them maybe have unique structures. So this slide, I just want to show you these small drugs and approved in 1981, 2019 for antiviral small molecules of antiviral drugs. If you look at the drugs, 75 small molecules have been proved during this probably 34 or 35 years. And if you look at the structures, many of them, actually 80% of them contain the common structure like purine, pyrimidine, and also like this glycinomide, quinidine. So the 80% of the structure actually almost all of them actually synthesized. But you see that our human imagination actually very limited. We can only based on this, you see this the similar pattern. So what we cause the problem is that because the similar structure of pattern, so probably they are easily to develop drug resistance. Okay, so not like natural products. If you look at natural products, I just show you that they are very complex structures. So this slide I also want to show you that the drug distribution. So when you pair with the synthetic structures and the natural products, and you compare them with the clinical use drugs, we find out the structure diversity pattern of natural products are very similar, actually very similar to the clinical drugs, but the synthetic drugs not very similar. There are few structure patterns. And so next time we're talking about our research. So our research is based on plant diversity of Lingnan region in China. So with Lingnan, so here just show you this Lingnan region. Lingnan region actually in southern part of the China. In the historically also the Lingnan region also include the northern part of Vietnam. Okay, so we focused on the plant in this area. And here, so show you the current the Lingnan region. So it's actually covered the several southern provinces for China, like Guangdong, Guangxi, and also including Hong Kong. So my current place in this region. So this region plant, the Lingnan region and the plant species are very diversified. In Hong Kong, even only in Hong Kong. So let's look at the Hong Kong. They contain this. The Hong Kong only 1,000 square kilometers. So contain, but actually more than 3,000 plant species, and also very strangely is that 2,100 plant species are native to Hong Kong. You cannot find them actually nearby like Shenzhen. So that's very strange. So for Lingnan region, actually more than 10,000 plant species are already discovered in this region. So, and we collect plants from this region, and we already collect more than like 6,000 plant extracts. We extracted them, made them a plant extract based on 2,000 species and 1,000 medicinal plant species. So with this plant extract library, so we can do a lot of work actually. So and to discover the antiviral compound from this plant extract, we actually developed antiviral evaluation system we call the one stone two birds evaluation system. So this slide just tell you that about this viral infection process. So we have this virus here. So if you look at this virus, how the virus invaded our cells. So there are actually 5 steps to the virus invaded us. So they attack. They called the viral attachment. The first step here, the viral attachment. They using the glycoprotein. They attach our cells and attach it here, and then they enter our cells and release the genetic stuff that's called and the viral entry. That's the second step. Then they did this RNA replication. That's the third step. Then they synthesize the proteins, the viral proteins. That's the fourth step. And at the fifth step, they assembled these proteins together. Then they brought it out from our cells. Then the new virus formed, and then they will start to replicate and to invade other cells. So that's how this virus infected. So these five steps actually we can design our drugs to target on these 5 steps. We can target on the like a viral attachment, the viral entry, also others like proteins, and also the mature rate. So so for our study, we focused on the viral entry. So for this first step and the second step. So that's so that's the next. So these are how we design our antiviral evaluation system. So we use glycoprotein and attach on use some virus core, so like HIV loop core. So this is not infected. Okay, so that's we take off those some of genetic stuff HIV. So it's not contagious. So this core can be handled in the biosafety 2 level lab. So we constructed some other viruses on the surface of this core. So then we they say H5N1 Ebola virus. So we can use the glycoproteins and coat it on the surface. And also we use the HIV look so that can light this virus. So then we incubated with the cells like human cells and incubated like A549. So then we measure this HIV look. Then we can measure this the signals. So the signals we can know how much. Then we treat with this with a drug and measure the cells, survive the cells, and we can know how much viruses can survive. So that's basically the principle for our antiviral evaluation system. I will not go for detail, but rather I will go and talk more about this later on our drug discovery. So here's the principle for our one stone two birds. With this one stone two birds antiviral evaluation system, we can actually detect the antiviral drugs at the same time. At least 2 or 3 antivirus. Like for the one setup of the assay, we can know that if the drug can be like an anti-H5N1 or anti-HIV or other virus like a SARS virus, so that we can do a lot of things with just like a simple setup for this assay. So here's this slide just show you this our how do we discover antiviral compound from the mixture of plants. So we first collect the plant material, small amount for probably several grams plant materials and make it as extracts. We make 1,000 extracts, then we screen them like HIV. So then once we got the active plant, that we go revisit the site. We collect the plants, more plant. We call the recollection of the active plant material. then we're doing bioassay guided fractionation, then we isolate the active compounds. Once we got active compounds, then we can do the structure modification, and we also can study further for this active compound, like to determine the mode of action. And so that's how we did our study based on this plant natural products. So next time we give you some example for my my study. Okay, so based on the plant diversity for Lingnan region. So we have actually screened more than 3,500 plant extracts using our one stone two birds antiviral evaluation system. So we also did the phytochemical study of 23 plant extracts, and using so-called antiviral bioassay guided fractionation. So then we isolated more than 50 active compounds, antiviral compounds, also more than 30 actually are new compounds. They have a novel structure. So I will give you several examples. So here is one example. This first example. So this we found this plant. So this urea nitida. It's actually traditional Chinese medicine. So and we discovered that can inhibit the H5N1 virus. So then we did the phytochemical study of this plant, and isolated a number of compounds from this plant, and one of them EN5. This one is a kind of flavonoid, and showed the anti-H5N1 activity with IC50 value about 1.86 micromolar. So we also did the structure modification of this compound. So I did not show the structure here. So here is another plant called Limia arbum, belong to Lamiaceae family. So this plant, we found that it shows the activity against the HCV, so HCV viral entry actually. Okay, we isolate the compounds called Limiridoxines A and B. So this compound is a new compound. So and it's a kind of actually is monoterpenes in chemistry, natural products we call them monoterpene. And this is active, so we found that showed this activity against the HCV with IC50 value about 2.331 micromolar, and the selective index is not toxic at 190 micromolar. So because the structure, so then we actually purchased more than dozens of similar structure, and evaluated this anti-HCV activity of this compound. Some of them actually actively showed similar anti-HCV activity with limiridoxine A and B. So, and also we did some structure modification for this. We didn't show here. And so here I show you this the same plants, the same plant, the water extract. We also found that this showed the anti-Ebola activity. So this you see it's 5 and 6. So this we treated the virus, anti-Ebola, the Ebola virus with water extract. They at 50 microgram per ml. All the actually the virus, the Ebola virus died. So this very interesting. So we also isolate a couple of compounds different from this limiridoxine. Actually, limiridoxine is not active against the Ebola, but the other compounds I didn't show here showed the activity against the Ebola. So. And this here is to show you another plant called Lijiang verticillata, also traditional Chinese medicine. So it belonging to Lauraceae family. So from this plant, we isolate actually two type of compound called sesquiterpenes. They have actually novel skeletons. They show the anti-HIV activity. So I isolated actually more than 40 compounds from these plants. Many of them actually show the anti-HIV activities. So also we further investigated additional Lichia plants in this genus. So this Lichia genus, and none for this Lichia plant species also showed antiviral activities. We're now doing further isolation of the chemicals from these plants. And here I show you another plant. It's called Vatica cinerea. So this plant also we discovered this plant has anti-HIV activity, and we did the phytochemical study. We did the bioassay guided fractionation study, and found that the chlorophyll. So this chlorophyll, one of the chlorophyll is called the fear-4 A5, showed very potent anti-HIV activity, and showed actually about 2 micromolar activity against HIV, and also is non-toxic at 20 microgram. So because this structure will further synthesized dozens of its structure derivative actually mimic the structure. So some of this compounds actually show the similar activity to this natural compound, and some of them actually show the more potent than the natural compound, the natural chlorophyll. So very interesting. So we are still try to figure out this mode of action for this chlorophyll against the virus. Okay. And so those are just some examples. We did the phytochemical study and structure modification, and we also did a further study of this plant extracts against the SARS virus, this new SARS virus. We evaluated actually thousands of plant extracts. Some of them actually we discovered some of them like edible plants actually are food can be like edible plant, medicinal plants are found that actually showed the inhibitory activity against the SARS coronavirus too. So like they are commonly the beverage like green tea. So we found a couple of tea different areas. We found some teas collected from Yunnan province in southwestern China. So this tea called poor tea is different from other tea. So this tea they have big leaves, not like other teas that small leaves, but this tea have big leaves. So this some of the compound, some of the gradients were isolated show that anti-SARS activity. So very interesting. We're still doing further study of this. Also we did some drug modification based on the gradients of A-beta-catechin and also T-flavin. So this flavonoids. Also, another discovery we found that the grape contains the resveratrol. We know is well known contain resveratrol. Some of the similar compounds we call the stabinoids also show the inhibitory effects against the SARS virus. So very interesting. So we also did the mechanism study, found that they are targeted on entry of this coronavirus. Very, very interesting. Also, they are very specific on target on the coronavirus, not other viruses. So these are just to show you our study. So we have evaluated thousands of plant extracts against the different viruses and many compounds. We have isolated many compounds and we evaluated these compounds. Also, many compounds showed antiviral activities against HIV, HCV, also HBV, and flu viruses, SARS, HSV. So these are just name a few. So these are viruses we are currently studying. Okay. So we also established a high throughput antivirus screening platform. So this can be used for screening against the different viruses. So SARS viruses, HIV, bird flu, even this high contagious like Ebola. So this just in biosafety 2 level. Not we do not need to use biosafety 3 or 4. So that's advantage of this screening system. So, and I will end off my topic, but I must give a lot of thanks to my colleagues, and not only in my university, also from other institutions. So like Dr. Harry Fang and Dr. Suyato, Dr. Zong, and Dr. New in Vietnam, and also this from University of Illinois Chicago, and also this work is also supported by my grants like Hong Kong also in NIH. So those are the financial support, so that's I can have this study. So, and also thanks my my group's people. So because of their efforts, so that we have this lot of compounds be isolated, also drug modified, also did the evaluation system, also did the mechanism study. So these are just some of the our group's people. So I will end up here. Thanks for your attention. Thank you. So hello, I'm
yes, thank you, Dr. Hongjie.
Yeah, thank you.
Yeah. Is there any questions from audience? Anyone?
So any question or
Yes.
have one? Well, you can show current slides for beta view. Oh, no questions. That's just
No questions.
Okay.
What type of challenges do you see in patenting?
You mean patenting? You asked a question, right?
Yes. No, I got a question from Dr. Don Turner. Can you just see the chart?
The charts. Okay. Which charts? Which charts?
No, chart. In this Zoom platform, you can see the chart option.
June chart. Wait, you mean this chart? No.
No.
Chart?
Chart box.
Which page?
No.
The plans.
There is a question from there. No. There is a question from Dr. Mr. Don Turner. It's what type of challenges do you see in patenting?
Can you hear me?
Yes.
Yeah, my question was, what type of challenges do you see in patenting, to formulate patents? Because I know oftentimes when you deal with natural products, it creates lots of problems when you try to patent.
Right. So yes, okay. The patents, because I just mentioned that we discovered the natural products from plants. So if they are novel structure, we can patent it in some countries, but not in the US, because US you cannot patent the natural products in the US because it's not immediate. But you can patent the novel natural products in in China, European, so other countries. But there is some solution we can do patent this compound. You do some drug modification, make it as a novel, so you can patent the compound. Also, there are some other ways you can patent the compound. You can patent the use, like this. For example, we just take example this paclitaxel. So this paclitaxel is used for anti-cancer. If we find this compound can be used against the virus, and this antiviral property has not been reported by other people, then you can also patent the use. So that's a different way you can do the patent filing. So.
Okay, thank you so much. One last question. So, and I joined late, forgive me. You may have covered this, but do you have any compounds that have proven to neutralize SARS-CoV-2?
Oh, yes. I just mentioned that in my here the slides. So Actually, we have discovered some compound from edible plants actually, and we used our assay. Our assay we call it one stone two birds, but it's actually you may call it a pseudo virus. And after we discovered the active compounds, then we send the active compound to another lab. They have this biosafety 3 and biosafety 4. They can test the compound against the live virus of SARS virus. Okay, so from their results, also our compounds are active against the live virus. So I didn't show you here because we have to get the agreement agrees to show the results, but they did tell me that some of my compounds really potent against this live SARS virus.
So did you actually get print 90 and print 50 data to measure the level of neutralization or SVNT? What type of test did you run?
It's neutral. Yes, they tell me that. They told me that. Yes. So in their lab, now I'm asking them because they also have animal models. So I'm asking them to if they can do some animal studies. So that would be probably can give a more convinced data. So